So in this section, we will look into different remaining milestones of this assembly pipeline, like overlapping, unit tagging. We'll talk about scaffolding, and we'll talk about this repeat resolution. So let's look into the overlapping first. So we got our sequences and we find those overlaps. We might allow some mismatches, so maybe like up to 5 to 6%. And then we do these overlaps. So once we have these overlaps, we can estimate, or we can have an idea of the true situation of these two genes in a chromosome. So it may be like something like this in the top case, or maybe something like this one. So we can have a repeat induced assembly, or we can have a no repeat kind of overlap situation. Next is we do unit tagging. So in unit tagging is basically derived from two words, uniquely assembled contigs. So we compute overlaps, consistent subassemblies, and we put these overlaps together, and there is small kind of you might have seen in your molecular biology courses something techniques called as chromosome walking. So by the help of these overlaps, you can assemble the larger fragments, and in the end we try to get a uniquely assembled contig. This is an illustration coming out from a software. How does this process look like in the software? In this diagram, we see that there is a unique DNA unitig, and we do have a unitig which is coming from a repetitive DNA. We can have a uniquely assembled contig from repeats, and we can have uniquely assembled contig coming from unique regions. So unitigs which are coming from repeats versus the coming from unique regions can be separated by using a process called as discriminator analysis or discrimination, where actually you discriminate between what is unique versus what is not unique. So the calculation is done as it's taken as a log ratio between the probabilities of the DNA being unique versus is repetitive. So here we are, for instance, putting a 2-copy DNA. So here we are getting a negative number. If we are getting a negative number, it's repetitive. If we are getting some positive number, so means this unitig is actually lying in this distribution, which is the distribution for unique, and here is the distribution for repeats, and there are overlaps obviously between these two distributions. Those small overlaps, actually they are smaller. So we are getting this negative and positive number since we are taking in this equation, we are taking the part being unique at the top. So probability of unique is here versus probability of duplicated or repetitive. So if this part is higher, it will give you negative number. This part is higher, it will give you a positive number. So when we plot these up two together, we can have these overlapping distributions. So anything that falls here is repetitive, and the value less than obviously this minus 10 or below that, and the values which are higher than 10, obviously they are unique, so they are lying in this distribution. The middle region is obviously where we are not sure about it. Next is scaffolding. So in scaffolding, we put those unitigs together, those unique unitigs with the help of paired end reads. So we rely on the reads which are paired end. So one of the member from this pair is in this one scaffold, like this guy, and the other one is in the second one bar. So we have 2 here and 2 bar here. So when they go and align with these different contigs, which are unique contigs, unitigs, we can look into the distance between them, and we can hypothesize that these two fragments, they can place together. But we need to confirm whether the distance is appropriate or not. Here is the diagram of the software. It's illustrating the scaffolds. The next is obviously repeat resolution. So for that, there are 3 different approaches. So number 1 is what you can do is pre-assembly. So you can find the regions which are repetitive. So you can find do some statistics and find what regions you might expect. And there is another database called as repeat database. So you can find those regions and mask them and maybe deal them separately. During the assembly, these repeats, they make up those tangles or some nets or meshworks. So you can detect them during the assembly also. In post assembly, you can find those regions, those repetitive regions and misassembly regions, and then in the end you can look into whether those, if you are using paired end reads while comparing their distances or the orientations, you can resolve these repeats. So overall what we have seen here is that the process is, this assembly process can be divided into these phases. Quality screening is an important factor, and obviously good overlaps are also a key to a good assembly, and we do unit tagging and we do scaffolding, and repeat resolution is a big challenge as far as these assemblies are concerned.